Hi, my name is Daniel, and today I would like to talk to you about the guidance cues for interneuron migration. Whether glutamatergic neurons develop in the dorsal telencephalon, which includes the cortex, the ganglionic eminences in the ventral telencephalon give rise to the GABAergic interneurons. Therefore, they need to first migrate into the cortex, where they can then integrate into the cortical circuitry. This migration process is called tangential migration, as opposed to the radial migration of the glutamatergic neurons during cortical layer formation. When we talked about cortical layer formation, we already encountered two migration modes, which were called somal translocation and locomotion. While extrinsic and intrinsic factors played an important role for both of them, locomotion also required radial glia as a guiding migration substrate. Compared to these two migration modes, tangential migration seems to be particularly challenging for multiple reasons. First, the interneurons must travel over very long distances. Second, they form multiple distinct migrational streams. And third, after reaching the cortex, they shift from tangential to a radial mode of migration to invade the cortical plate. Today, I would like to use the cortical interneurons as an example to show you how extrinsic guidance cues, intrinsic factors, and the migration substrate work together to enable such a complex process. Let's start with the extrinsic cues. Interneurons utilize gradients of secreted chemoattractive and repulsive cues to migrate towards the cortex. One key chemokine is stromal-derived factor 1, also known as CXCL12, which gets expressed and secreted in the marginal zone and intermediate zone of the cortex. This extrinsic signal attracts interneurons expressing the corresponding receptor CXCR4 and plays an important role in defining marginal zone and intermediate zone as the main migrational paths for interneurons. Neuregulin 1, with its receptor ERB4, is another such chemoattractant. At the other end, the ganglionic eminence secretes the chemorepulsive cue afrin A5, which initiates the interneuron migration. While these cues already provide the right direction, there is another repulsive signal coming from the developing striatum, semaphorin 3A and 3F. It prevents the neuropilin-expressing cortical interneurons from migrating into the striatum and thus helps confine them to distinct tangential streams. So now that we've understood how multiple migrational streams are formed, let's talk about the role of intrinsic factors. The spatiotemporal origin of interneurons within the ventral telencephalon does not only determine their subtype, but also pre-specifies their migratory routes. This intrinsic migratory fate is specified by the expression of different key transcription factors, for example NKX2.1, and receptor patterns, enabling the migrating cells to respond selectively to route-specific environmental cues. One example are the striatal interneurons, which do not express the receptor for the repulsive semaphorin signal, allowing them to migrate into the developing striatum. This birth date-dependent migratory fate goes so far that interneurons and projection neurons born at the same time often reside in the same cortical layers. However, this initial transcriptional program and receptor pattern changes as the migrating interneurons age on their way. When the interneurons arrive in the cortex, they lose their CXCR4 receptors, and thus their attraction to the STF1 external cue, which marks the beginning of their radial invasion into the cortical plate. This explains how dynamic transcriptional programs can specify the intrinsic migratory fate and coordinate something like a migratory shift. The last question that remains is how these migratory signals can be maintained over such a long distance, and if there is a supporting migration substrate. One helpful thing to know is that the chemoattractive neuregulin 1 does not only get secreted in the cortex, there is also a membrane-bound isoform. This allows neuregulin 1 to also act as a short-range attractant, creating a permissive corridor for migrating interneurons. It is known that cell-cell interactions and cell adhesion molecules play an important role in tangential migration, and migrating interneurons have been reported to contact corticofugal axons, which might be used as a scaffold for migration. Even stage-dependent substrate preferences have been discussed, which gives us a better idea of how migration over such long distances can be realized.